Hello friends, welcome to our channel Allied Health Educare. Allied Health Educare is the fastest growing online coaching center that provide good quality of MLT learning. So we are representing several type of important and specific topic by which you can get a good idea about what you are doing in your everyday lab. So today we have come we have come up with a new topic, that is CBC report interpretation. What is CBC? You all may know what is CBC. CBC stands for complete blood count. By doing complete blood count, we get a clear picture about our cell count, different type of RBC indices like MCV, MCH, MCAC, RDW, etc. So we may face difficulties in case of CBC report interpretation. The CBC report interpretation in terms of anemia, which appears in the lecture, so that you can get a good idea how to interpret the CBC report. So I have prepared different slides which explains how you can report, how you can explain your CBC report. So let's move to the our first slide. The first slide depicts anemia. What is anemia? You all may know what is anemia. Anemia means the deficiency of hemoglobin. When hemoglobin value goes below 13 gram per day in case of male, and in case of female, when hemoglobin goes below 12 mg per dl, then it is termed as anemia. There are different type of classification of anemia in terms of morphological classification and in terms of etiological classification. So in morphological classification, there are three types: microcytic, normocytic, and macrocytic. And on the other hand, etiological classification depicts three types of classification, that is hemorrhagic, second one is dyshematopoietic, and third is hemolytic. So you have an idea what is anemia, what is RBC count, what is WBC count, how to measure hemoglobin, but you may don't have any idea about CBC report interpretation. In every day in our lab, we run many CBC samples. In CBC sample, some sample yields normal result, some sample gives abnormal result. So now we are learning, we are going to learning, and I am going to teach you how to interpret this type of abnormal report. So first of all, before proceeding to any abnormal report, you must have an idea about normal sample. If you don't have any idea about normal sample, then how can you understand the abnormal sample, abnormal report? So here is a blood picture. This is the blood picture, and this is the corresponding CBC report. There are different parameters: hemoglobin, RBC, BCV, MCV, MCH, MCEC, and RDW. So in this picture, you can see the RBC morphology as normal. How you can interpret it is normal? So this is the RBC. So in this picture, you can see I have drawn three RBC. First one is normal. First one is termed as normal RBC. Second one is hypochromic RBC, and third one is severe hypochromic RBC. Normal RBC. What is normal RBC? Normal RBC means this RBC has sufficient content of hemoglobin. That is normal RBC. How can you determine whether it is normal or abnormal? Look at the center. look at the center of the RBC, you can see there is a white discoloration. This is called central pallor. The length of the central pallor is 1/3 of the diameter. The central pallor is 1/3 of the diameter. If the diameter is less than or equals to 1/3, then the RBC is okay, and it is called normocytic RBC. Normo means normal, and cytic means site or cell. Next, move to the second picture. Here, the central pallor region is much more broader, and it is obviously greater than 1/3 of the diameter. So hypochromic RBC, in this picture, from this picture, we can understand that in case of hypochromic RBC, there is a deficiency of hemoglobin in RBC. So it is called hypochromic RBC. And in case of hypochromic RBC, the central pallor must be greater than 1/3 of the diameter. The central pallor must be greater than 1/3 of the diameter. And in case of severe hypochromic, on the other hand, in case of we can see this picture, there is a severe hypochromia. Severe hypochromia means the central pallor is much more broader as compared to hypochromic RBC. So this is the general picture of a RBC, how a RBC looks like under the microscope. And this is the peripheral blood smear. So peripheral blood smear looks absolutely normal. And if we compare the peripheral blood smear with the CBC report, the hemoglobin is 15. Hemoglobin is 15 means it is normal absolutely. RBC is 4.6 million. That is also normal. PCV is 43%. MCV is 84 femtoliter. MCH is 29.3 picogram. MCHC 33%, and RDW is 12.5%. So all parameter is normal. Now, what is hemoglobin? You may know that what is hemoglobin. Hemoglobin is a protein. It is a quaternary protein that helps in transportation of oxygen from the lung to the different type of tissue. And RBC count is absolutely normal. It is within the normal range. And PCV means packed cell volume is 43. You know that what is PCV? Packed cell volume. Packed cell volume is also called hematocrit. And mid corpuscular volume, MCV is 84 femtoliter. 84 femtoliter means the volume of RBC. The volume of RBC within the normal range. If it less than 80 femtoliter, then it is termed as microcytic RBC. And here is the MCH. MCH is the greater than 26. If it become less than 26, then it is termed as hypochromic RBC. So in this picture, it is determined that the blood picture is absolutely normal. So there is no problem at all. Now move to the next slide. This is the next slide. Here, what we see, the hemoglobin is low. The hemoglobin is low. First, look at the peripheral blood smear. Peripheral blood smear looks absolutely normal because the central pallor is absolutely normal. The central pallor is this is central pallor. This central pallor looks absolutely normal because it is within the normal range, that is 1/3 of the total diameter. The main problem in this blood picture is RBC count is low. The RBC count is low. RBC count is 3.5 million. So it is absolutely low count of RBC. But if we see the RBC indices being MCV, MCH, MCHC, these three parameters are absolutely normal. PCV is also low. RBC count low. If RBC count is low, then it is obvious that PCV count will also be low, and hemoglobin is also low, that is 10.5, that is less than 13. But the interesting thing is MCV, MCH, and MCHC is absolutely normal. What does it mean? What does it mean? It means that the production of RBC from the bone marrow is absolutely normal. Now we can find out the what is the reason behind the deficiency of RBC and why RBC become low. The main problem is if some people meet an accident, then all the blood comes out his body. Then what happens? The patient may become hypovolemic, so that the RBC count is also low, PCP will become low, and hemoglobin will become low. But the RBC increases will become normal because RBC production is normal. There is no problem in RBC due to the some reason blood loss is there. Due to the blood loss, the hemoglobin value will become low, RBC and PCP will become low. But if, as the RBC production is normal, there is no problem in RBC production, then the MCV, MCH, and MCEC is absolutely normal, and as well as the hair cell distribution with I will later discuss what is RTW 12. So all become normal. So it means that this anemia is called or is termed as normocytic normochromic anemia. What is anemia? Because the hemoglobin value is low. So the hemoglobin value is low, then it is definitely it is anemia. But what is normocytic? Why it is called normocytic? Normocytic means the volume of RBC, mean corpuscular volume is RBC is normal. That is normocytic. And why it is called normochromic? Normochromic means the mean corpuscular hemoglobin is absolutely normal. So that is why it is called normocytic and normochromic anemia. In which cases we can see this type of anemia, severe hemorrhagic condition, severe blood loss. In two, three reasons, we can see that this type of uh, result we make it. Now, next move to the next slide. Another one is very very important slide. This is a very very important slide because you may encounter this type of result in your everyday lab work. In this picture, in this previous, first look at the previous peripheral blood smear picture. What you have seen that the RBC, first look at the RBC. What you see? What you see from the picture? The central pallor, what I have said earlier, the central pallor is much more broader in comparison to normal RBC. You look at the RBC, you can understand the central pallor is much more broader. The central pallor much more broader. What does it mean? It means the RBC is hypochromic. The hemoglobin per RBC is low. That is why it is called hypochromic. So if we compare this blood picture with this CBC report, we may find that hemoglobin is 8.1. 8.1 means it is low hemoglobin value. It is obviously and definitely it is anemic patient. RBC count is low, 3.2. PCV is also low. MCV is less than 80 femtoliter. If MCV is less than 80 femtoliter, then it is called hypochromic. MCH is less than 26 picogram, so that it is hypochromic. And if MCH or MCB is low, then MCAC definitely low. That is 29. It is also low. But RDW. Now, what is RDW? RDW stands for red shell distribution width. What does it mean? What does it mean? RDW. RDW means there is a variation. If RDW is high, it means there is a variation between the RBC sizes. I'm showing you two pictures. Look at this picture very carefully. I'm showing you different, hypothetically assume it is a blood smear, the PBS, peripheral blood smear picture. Here is the RBC. This is the blood picture. It is sample 1. And this blood picture is for sample 2. If we compare two blood picture, two peripheral blood smear picture, it is very obvious that in case of sample 2, there is a variation in RBC size. In case of sample 1, the RBC sizes is uh, is same, or there is a little variation. But in case of sample 2, there is a much more variation between the RBC sizes. This variation between the sizes of RBC is called anisocytosis. What is called? What is anisocytosis? Anisocytosis means variation between the RBC size. That is called anisocytosis. And anisocytosis represent RDW. More the variation between the RBC size, more the RDW value. So here, the normal range of RDW lies between 11% to 14%. Actually, this type of RDW is it's expressed as red shell distribution with CV percentage. CV percentage. So that is why we have to express the RDW value in percentage. So here the RDW value is greater than 14% or at least 22.5%, means there is a great variation between the RBC sizes. And most common thing is, if we find the RDW 22.5%, as I have earlier told, it is called anisocytosis. Another thing is, if there is a variation between the shape of RBC, variation between the shape of RBC, that is called poikilocytosis. The poikilocytosis means, what is poikilocytosis? What is poikilocytosis? Poikilocytosis means variation between the RBC shapes, not size. And if anisocytosis and poikilocytosis coexist, then it is called anisopoikilocytosis. So this anisopoikilocytosis is evident in case of iron deficiency anemia. Actually, this blood picture that is given here, it is a microcytic hypochromic RBC, and there is anisopoikilocytosis. Different shape of RBC may be seen here. This is teardrop cell, this is elliptocyte, and you may see that there is a variation in shape also, and as well as size also. So here in this picture, anisopoikilocytosis is very much evident. In case of iron deficiency anemia, we can get this type of blood picture. So in case of iron deficiency anemia, it is very important to remember. It is very important to remember, in case of iron deficiency anemia, MCV must be less than 80 femtoliter. In case of iron deficiency anemia, MCV must be less than 80 femtoliter, and MCIC must be less than 26 picogram. And RDW must be greater than 14%. Three parameter. The abnormality in three parameter represent the iron deficiency anemia. So Whether it is iron deficiency anemia, you have to confirm it before releasing the report. You may give a note when you release the report in CBC report. Then you may write in comment that ferritin or iron profile must be done. If it is iron deficiency anemia, then the ferritin must be below the normal range. The ferritin is the one of the most confirmatory tests in case of iron deficiency anemia. In iron deficiency anemia, the ferritin must be below the normal range. And if it is iron deficiency anemia, the other parameter, that is serum iron will also be low. Total iron binding capacity will increases. So this is the very important corresponding test that confirms it is a iron deficiency anemia. So in case of iron deficiency anemia, to identify iron deficiency anemia very precisely, you have to look into the peripheral blood smear as well as the MCV value, and obviously hemoglobin value. Hemoglobin value you have to see it at first. Hemoglobin value you have to see at first. Then you may proceed to MCV value, then MCH, and lastly RDW. Next, move to the another PBS. So here, what what is seen? Hemoglobin is low, RBC is low as well, hematocrit is low as well, MCV is normal, MCHC is normal, MCHC is slightly high or normal, and RDW is severely high, and there is an increase in reticulocyte count in the sample. If we look into very carefully peripheral blood smear, you may find out this type of RBC which does not have any central pallor. This RBC, I am pointing it with a black arrow. This RBC has central pallor. This RBC has central pallor, but this red marked or red arrow marked RBC does not have any central pallor. These RBCs are called spherocyte. These RBCs are called spherocyte, and this is called hereditary spherocytic anemia. A hereditary spherocytosis. What is hereditary spherocytosis? Hereditary spherocytosis means there is a problem in cytoskeleton of RBC. There are different type of protein in the cytoskeleton of RBC which control the flexibility of RBC. RBC is a very flexible cell because the RBC has to pass through the very narrow blood vessels or blood venules. So it has to maintain the flexibility. But where from its flexibility the RBC may get? There are different type of protein, ankylin, spectin. They can control the flexibility, or they may provide the flexibility to the RBC. If there is a deficiency of this type of, it is obviously it is a genetic problem. If there is a deficiency of this type of protein, then the RBC loses its flexibility. As we know that there is a filtration process in our body. There is a tiny organ that is called spleen. The spleen filters blood. If spleen encounter any abnormal type of RBC, then it eliminates the RBC. So in case of spherocytosis, hereditary spherocytosis, when the RBC loses its flexibility, then it becomes spherical, just like a football. The RBC, the shape of RBC is actually coin shaped. It is a coin, like a 5 rupee coin, and it is also biconcave shape. If it become spherical, then it become just like a football. So if the spleen encountered this type of abnormal RBC, then it eliminates the RBC. And this elimination of the RBC by the splenic macrophage will leads to anemia. And this type of anemia is called hereditary spherocytosis by hereditary spherocytic anemia. And it is a one special type of hemolytic anemia, extravascular hemolytic anemia. That is why reticulocyte count goes up. Normal count lies between 1% to 2%, but when it becomes spherical and it results in the spherical shape. The spherical shape of the RBC results in the elimination of RBC, and the elimination of RBC leads to the increased value of reticulocyte. So, if reticulocyte count is high, then you can understand that this is a hemolytic anemia, or you can interpret one type of hemolytic anemia. So, what happened? In case of hemolytic anemia, there are different type of hemolytic anemia: autoimmune hemolytic anemia, drug-induced hemolytic anemia, sickle cell anemia, disease-free deficiency anemia. One thing you have to remember: if it is anemia, hemoglobin must be low, RBC count must be low, PCB count must be low. But if it is hemolytic anemia, then the RBC indices become normal, or it become slightly, very, very slightly low. What we have seen previously, in case of iron deficiency anemia, MCB value is much more below the 80, MCH value much more below the 26 picogram, and in case of hereditary spherocytosis, obviously it is a hemolytic anemia. In case of hemolytic anemia, it becomes normal. So you can see the corresponding report: the MCH normal, MCV normal, and MCH also normal. So clear picture of it is a clear picture of hereditary spherocytosis. One thing must be remembered: merely seeing the CBC report, you cannot interpret the nature of anemia. You have to see the peripheral blood smear as well. Without seeing the peripheral blood smear, you cannot interpret whether it is hereditary spherocytosis or another. Now move to the next thing. It is very interesting. You may have encountered this type of report several times in your lab. So hemoglobin is as usual low. RBC count is also low. PCB value is low, below 45%. But very, very interesting thing: MCB value is greater than 100 femtoliter. The normal value of MCB is 80 to 100 femtoliter. But here, the MCB value is greater than 100 femtoliter. It means there is hypochromic RBC. So not hypochromic, macrocytic RBC. Please pardon me. This is my macrocytic RBC. We have previously seen that if MCB below 80, it is called microcytic RBC. If the MCB value goes beyond 100 femtoliter, then it is called macrocytic RBC. So there are two blood picture. Here you can see that the RBC size is slightly larger. The volume of RBC is slightly larger as compared to normal RBC. And this is called macrocytic anemia. If the hemoglobin value is low and MCB value is greater than 100 femtoliter, then it is called macrocytic anemia. And in case of macrocytic anemia, MCH value is greater and MCHC value is greater or not. This is a very important situation in case of macrocytic anemia. The MCV is greater than 100 femtoliter and MCH become 39.6. If the RBC size is bigger, then much more hemoglobin is inside in the RBC. And here RDW is quite normal. So this is called macrocytic anemia, and it is in this picture from this peripheral blood smear we can one thing we can interpret that it is obviously hyperchromic, not hypochromic, hyperchromic. Pardon, hyperchromic. In case of vitamin B12 deficiency and folic acid deficiency, this type of anemia can be caused. So vitamin B12 deficiency or vitamin B9 deficiency leads to the inhibition of DNA synthesis. If there is a vitamin B12 or vitamin B9 lacking, it leads to the inhibition of DNA synthesis, or there is a impaired DNA synthesis. This impaired DNA synthesis causes RBC size to get bigger. So this is called macrocytic anemia. And in case of macrocytic anemia, one of the most important or characteristic features is hyper-segmented neutrophil. In case of normal neutrophil, you may find maximum 3 to 5 lobes, nuclear lobes. You know that nucleus of the neutrophil is lobular nucleus. So there are 3 to 5 maximum lobe may be present. In case of macrocytic anemia, if the macrocytic anemia caused by vitamin B12 deficiency or B9 deficiency, you may find the hyper-segmented neutrophil. Hyper-segmented neutrophil means the lobe of neutrophil, the lobes of neutrophil may be greater than 6. This is called hyper-segmented neutrophil. You can see from the picture, this is a hyper-segmented neutrophil, and this is also a hyper-segmented neutrophil. So it is very evident from the blood smear picture and as well from the CBC report, we can understand it is a macrocytic anemia. You have to just look two things. Look into the two things. Another one, first one is hemoglobin value, and second one is MCV value. If we find that the hemoglobin value is low and MCV value is high, greater than 100 femtoliter, then you can interpret the report as a macrocytic anemia, and then you have to check out the vitamin B12 deficit, vitamin B12 value. What is the value of vitamin B12 in our serum or vitamin B9 deficiency? We know that vitamin B12 and vitamin B12 and vitamin B9, they are dependent on each other. If there is a vitamin B12 deficiency, there is automatically vitamin B9 deficiency, and vice versa. If there is a vitamin B9 deficiency, then it automatically there is a vitamin B12 deficiency. So you may check the value of vitamin B12 and vitamin B9. You may find that vitamin B12 and vitamin B9, the value of serum vitamin B9 and vitamin B12 is much more lower than the normal reference range. The next interesting thing is this type of blood picture. By merely seeing in the blood picture, you may say that this is a blood picture of a thalassemia patient. Yes, it is. You are absolutely correct. How can you interpret it is a thalassemia patient? There are several types. There are several, but even there are numerous podocyte. These cells are podocyte. I am pointing it out with a red arrow. These are podocyte. These are called podocyte. So podocyte is the very characteristics and interesting features of thalassemia. Actually, this is the picture of thalassemia. That picture of a thalassemic patient. It is a 9 years old boy. So here hemoglobin is low, PCB is all. I mean, I am coming to the RBC point next letter. PCB is low, MCB is low, that is less than 80 femtoliter, and MCH is low, that is less than 26 picogram, and MCH also low, and there is a slightly increase of RTW, slightly increase. Very interesting features. But in relation to HB hemoglobin value, in relation to hemoglobin value, RBC value is much more higher. Here the RBC value must be 3 to 4 million. But here the RBC value is, if we compare the RBC value with hemoglobin, it is much more higher, that is 5.6. This is the very important features of thalassemia trait. Thalassemia trait is called thalassemia carrier. In case of thalassemia carrier, the hemoglobin must be low, PCB must be low, MCB must be below 80 femtoliter, MCHC below the 26 picogram, and RDW may be normal or it may be slightly increased. But the most important features of the thalassemia trait is high RBC count. You must remember this point. In case of beta thalassemia trait, in case of beta thalassemia trait, RBC value is much more higher as compared to hemoglobin value. Here hemoglobin value is 10.5, the RBC value must be around 3.5 to 4, but here is 5.6. That means RBC is slightly higher as compared to hemoglobin. If we compare the RBC count value with hemoglobin. So in this blood picture, another thing is we are observing different or numerous chorocyte. Chorocyte means target cell. So this picture is self-explanatory, and it can be determined from this CBC and as well as the blood picture. It is a blood picture of thalassemia carrier. Now it is the exercise time. So this is the blood picture, this is the PBS, and this is the CBC parameter and it is value. So here we can see that hemoglobin value is low, RBC value is slightly low, PCB value low, MCB value less than 80, MCH value less than 26, and RDW value is greater than 14%. And there is a microcytic hypochromic RBC, and there is anisocoagulocytosis exist. So we can interpret it as a iron deficiency anemia. So in this blood picture, we can see very clearly there are different type of abnormal cell. This is the abnormal cell. The cell is just like sickle cell. The cell is just like sickle cell. This it looks like this type. And there are numerous target cells. There are numerous target cells. In CBC report, in CBC parameter, hemoglobin is low, RBC is low, PCB is low, MCB is normal, MCH is normal, MCHC is normal, and RDW is high. From the previous slide, we learned that if hemoglobin is low, RBC is low, but when the MCV, MCH or MCHC, the three RBC indices remains normal, then it strongly indicates towards the hemolytic anemia. So it is actually the blood picture of a person is suffering from sickle cell anemia. But from the CBC parameter or CBC report, we cannot understand whether it is sickle cell anemia or different type of hemolytic anemia. So from the blood picture, we may find you may find several sickle cell or sickle cell RBC. It is a sickle cell anemia. You have to confirm the sickle cell anemia by doing hemoglobin electrophoresis or HPLC. Here is the HPLC report. Electrophoresis. You can see this is the hemoglobin electrophoresis report, and here the hemoglobin S is 84.5%, and it is an evident case of sickle cell homozygous. It is a sickle cell homozygous. So in case of sickle cell anemia, you may not found any type of sickle cell in the blood picture. Then you have to confirm. You have to confirm it by doing hemoglobin electrophoresis. The CBC report cannot reflect whether it is sickle cell anemia or not. So you have to rely on two things. One is peripheral blood smear, and you may do some other test that can determine or that can confirm that it is a sickle cell or sickle cell anemia. What happened in sickle cell anemia? In case of sickle cell anemia, it is also is a genetic. There is a different type of genetic reason. That is why there is a shifting of uh, amino acid in beta chain of hemoglobin, and that leads to the formation of abnormal hemoglobin. And this abnormal hemoglobin causes polymerization, and this polymerization of hemoglobin affects the shape of the RBC, and the RBC become sickle cell. And this sickle cell will appear when the hemoglobin become deoxygenated. Once the hemoglobin become deoxygenated, so the hemoglobin polymerize and it becomes sickle cell. So here in the blood picture, uh, here we see the 3 to 4 or 5, 6, 5 to 6 sickle cell. That means the hemoglobin in this RBC is deoxidized. The hemoglobin is deoxidized. So in case of when the hemoglobin releases the oxygen, it become polymerized and it may alter the shape of the RBC. So when the hemoglobin becomes deoxygenated, then this sickle cell can appear in the blood picture. Next, last on today's in our lecture, by doing this type of blood picture, I can conclude my lecture. So here, what we have seen, what you have seen here. First, look at the CBC report. Hemoglobin is low, RBC is low too, PCB is low, MCB is slightly low. It is considered to be normal. MCH also is normal, and MCHC is normal, but RDW is slightly high. The value of RDW is slightly high. So when we see the blood picture, very interesting cells are seen. This type of cell. This type of cells here. This is called bite cell. This is called bite cell. And as we learned earlier, if the hemoglobin is low, RBC count is low, but RBC indices are normal, it indicates towards the hemolytic anemia. So here in the picture, we see that the RBC count is low, and here is special type of RBC. This is called bite cell. This bite cell is the special, interesting or characteristic features of G6PD deficiency anemia. We know that G6PD is an enzyme, glucose 6-phosphate dehydrogenase. It is an enzyme. It is an enzyme that helps produce NADPH. NADPH is an antioxidant in our body, which inhibit different type of free radicals or reactive oxygen species, and thereby protecting different cell from being oxidized. So if the G6PD will become low in our body, if G6PD enzyme is low in our body, then it leads to the inhibition of or down regulation of hexose monophosphate pathway. Hexose monophosphate pathway will be down regulated due to the deficiency of glucose 6-phosphate dehydrogenase. And we all know that hexose monophosphate pathway produces NADPH. This hexose monophosphate pathway will produce NADPH, which inhibits different free radicals, superoxide, and reactive and other reactive oxygen species. If hexose monophosphate pathway is downregulated due to the deficiency or lacking of the G6PD enzyme, then NADPH also become low, and this results in increase in oxidative stress in our body. If oxidative stress increases in our body, then different organ or different cell may be affected. As for example, RBC membrane, our retina, our cell membrane may be affected or it become oxidized. So it become, if the RBC membrane is become oxidized, so what happened? Our hemoglobin will become oxidized by ROS. And if this hemoglobin, if the hemoglobin is oxidized, then it is called methemoglobin. This is called methemoglobin. This methemoglobin is oxidized form of hemoglobin. Oxidized form of hemoglobin. In hemoglobin, the Fe is double plus. If it is oxidized, then it loses one electron and it becomes Fe plus 3. So this methemoglobin is non-functional and it tends to deposit in the RBC. It tends to deposit in the RBC. And this deposited methemoglobin is called Heinz body. This deposited methemoglobin is called Heinz body. Heinz body cannot be demonstrated by any kind of Romanowski stain, Leishman stain, etc. This type of Heinz body can be demonstrated by supravital stain. Here in this picture, you can see the RBC contains Heinz body. So this Heinz body is an abnormal thing in our RBC. Earlier I have told that uh, in our body there is a minute organ that is called spleen. Spleen filter our blood. When the spleen encounter this type of RBC, assume this is the RBC. and this RBC is having Heinz body. Heinz body is a round shape, and it is just look like that. If the RBC with Heinz body encountered by spleen, let's assume this is our spleen. So what spleen has does? Spleen has macrophage. This is called splenic macrophage. Splenic macrophage phagocytose this portion. Phagocytose this portion. So after phagocytosis, the RBC become like that. So the macrophage remove this portion. The macrophage of spleen, what they have done, they have removed this portion. So the RBC shape is just look like a beaten shape. So this is called bite cell. This is called bite cell. So furthermore, we have to do our we have to check, or we have to determine the G6PD level in our blood. If it is G6PD deficiency anemia, so the G6PD value goes below the normal reference range. So by this process, we can identify this blood or this patient is suffering from G6PD deficiency anemia, or this patient have G6PD deficiency. So by merely seeing the blood smear and as well as by seeing the CBC report, we can ourselves can interpret different type of situation or different type of condition. We don't have to rely on the physicist, or we don't need to rely on the doctor what they have reported. We can do report, we can do with medical technologies, can do report ourselves. And this is the most important part of learning. So I am concluding my lecture. This lecture is part one. Allied Health Educare is providing you much more interesting video, and we also we also come up with new lecture part two. So stay tuned to our channel, share, subscribe with your friends, and please support us so that I can make this type of video in future. So thank you all. Very good night.